All right. Um, cool. So, welcome to Journal Club. Today, I'm going to be talking about a paper called "Lateral Gene Transfer Generates Accessory Genes That Accumulate at Different Rates Within a Grass Lineage." And then I put a little subtitle there, called it "Reproductive Contamination Through Illegitimate Pollination," because that is by far my favorite line from the entire paper. Don't worry, I'm going to explain what all those pieces of the sentence mean. So the key points in this paper, which, by the way, should be available as a link in the Journal Club channel. So lateral gene transfer is a movement of DNA between organisms without sexual reproduction. The acquired genes represent genetic novelties that have independently evolved in the donor's genome. Now, by the way, if it sounds like I'm repeating straight from some of the key points in the paper, I absolutely am, because they spent a lot of time making these make sense. So I'm not really, this isn't a copyright issue. Like, these are their words. Phylogenetic genetic methods have shown that lateral gene transfer is widespread across the entire grass family, although we know little about the underlying dynamics. And then it says, see other materials. And so this sentence right here, that is widespread across the entire grass family, the supplemental material of this paper gets into this, and we've got a slide about that. And it's actually very, very cool. So the other points go on to just talk about like how they basically did it. They got, 5 key points for a certain type of grass. And then they got further what they're called like accession individuals, which are just basically like tiny little budding branches. They're not, we're not speciating here. But you're definitely starting to show a collection of genes that are hyper specific to like you, your kin, and the region that you're in. And then they found 168 laterally acquired genes, which is a lot. And there's a whole chunk of the paper where they talk about how extremely conservative the model is, how much dead space there is in data, which I really great. They actually go really in depth on all these things. This paper is actually really fun because you can get excited about it for a lot of different reasons. And then this last one here about the turnover rate and how this is like an evolutionary trait, and so we're going to kind of talk about all of those things. But just for a definition of terms before I see if there are any questions, there's this concept that he's talking about called lateral gene transfer. Okay, and so lateral gene transfer is the movement of genetic material between organisms, like other than the standard parent-offspring model. And yes, we're not talking about like, and yes, that means whatever parent-offspring model you have for the species. So yes, budding, for instance, is vertical. And it's an important adaptive force in evolution, contributing to metabolic, physiological, and ecological innovation in most prokaryotes and some eukaryotes. And the whole like it being a thing in eukaryotes has it's something that people were a little contentious with for a while. But it's not really something that we have the ability to argue with anymore. And papers like this are exactly why. And so down here you see that I've written transformation, transduction, and conjugation. Does anyone want to take a shot at what those terms are there for, like what they mean? Okay, so these are the 3 primary methods for lateral gene transfer. So transformation is just the absorption of outside DNA. And so we do transformation like when we add a plasmid to a bacteria. That's a cellular transformation, right? We're adding in the genes, transforming it. Transduction is when it happens with a virus. So when we use AAV to do a genetic therapy, that's a transduction. And then conjugation is literally like two cells smashed into each other, and it's, it got messy. Like literally just like garbage bleed over. There's like instances where this happens where like in between the rooting system, and they could actually map out where the genes are and how they move through the plant, and they're like, yep, it looks like it happens just because the roots just keep bonking into each other. That is not, however, the reason that they decided that this was all happening in grass. They were going with that charming quote about pollen. So they think it's basically just like pollen doing things that it's not like, that we don't necessarily talk about pollen doing. So lateral gene transfer, just in case anyone thought this sounds familiar, is like horizontal gene transfer. It's like the same thing. We just kind of have stopped saying horizontal gene transfer. I'm pretty sure it's because like GMOs scare people, like to freak out about that concept of like genes jumping in between species. And there's no real like good way to turn around and be like, dude, you don't know the half of it. Like it gets pretty sloppy down there on the cellular level. But yeah, so before I go any further, does anyone have any questions so far?
Yeah, so I was wondering about the conjugation thing. That happens with like single-celled stuff, but you mentioned pollen, so like is pollen
No,
the
it
same?
can totally happen in eukaryotes. Like
Oh,
those are the methods,
okay.
and they work more commonly in prokaryotes because they got a whole different chassis, a completely different hardware set that they're working with. But like there are some fundamental similarities, and in those areas, the avenue for change is always explored.
Okay.
Mhm.
Gotcha.
I have two questions.
Sure.
One, would the capture of organelles be considered horizontal gene transfer? Like, like
Like the whole mitochondria thing.
yeah, and then yeah, basically yeah.
Yeah, I mean, if you have a chunk of genetic material that is coming from another species, and you integrate that to the degree that it is maintained throughout successful generations, and don't worry, we do talk about this. Like it also has to do with how long it stays, and like the amount of like it's got to be. There's a whole hierarchy of where the genes come from, how they got there, and what pressures are exerted on them that maintains like rates of change and adoption and decay as well. So
Cool.
yeah,
And I think I've heard about this before with that one really basal angiosperm.
one really big.
It's not a water lily. It's even more basal than that. It's from
Like a
New
crest or
Caledonia.
something. Okay.
It's from it's the 1 second. It's its own clade or own let me see. Basal angiosperm, because they found like moss in it. Moss genes. Amberella. Yeah. Yeah.
Yeah, with moss donors. Very cool.
Yeah. Right.
Yeah. So that stuff happens a lot. And and so this is the point where I actually, before I even got super into the rest of the paper, which there's still a lot to go, there's some other papers that I suggest if anyone wants to like learn more about lateral gene transfer in eukaryotes, and that whole mechanisms underpinning lateral gene transfer between grasses, which is also Dunning. Like they reference Dunning a lot, and that makes sense because the paper is written by Dunning. But this is like their field, this whole thing, and part of this is, as we're going to see, goes into well, the technological advancement that they're kind of at the forefront of. So at the beginning of the paper, before they even started talking about how they did everything else, they're like, look, here are the 5 different things that everybody likes to talk about. Whenever we're talking about lateral gene transfer in eukaryotes, everyone's like, here's our bucket list of why your data is not conclusive enough, or, well, what about this? Or what about this? And so they went through and they were just like, this is their bullet point list again. And they're just like, look, here are all the problems. Here are all the reasons it's not a problem for this situation. And in the paper, they like made it in like a little cutty pasty type of thing. They're like, yep, we want you to be able to take this and cut it and paste it. And I'm like, absolutely, that's a great idea. But so you can see that a lot of our a lot of the alternative hypotheses that we have actually have to do a lot with our poor control of the technology, or our lack of understanding of the technology, or our just like shitty PPE. And so because we try to do a lot of gene transfer, right? I mean, like we're explicitly like in, you know, most of our genetic engineering is being done with like our aggressive attempts at lateral gene transfer. We're not like out there sitting rubbing 2 grass seeds together hoping we're going to get more vitamin A or something in some rice. Yes, I know that's not how grass have sex. But yeah, so these are all the things that they covered on that. Does anyone, if anybody wants to come back to this list, put a pin in it and let me know at the end, because each one of these are kind of like their own topic about the type of stuff that happens, you know, in the field. So they had a fairly simple method for this, and the most like when you look at the method, it's extremely straightforward. They're like, okay, we're setting up sample genomes. We've got 5 reference points, and so they found like these multiple species spread out over the over the space of kind of like the same type of biome. So it like cuts across the top of Africa and the lower part of China. And then they did full sequence data, and they found 40 additional accessions on top of these, which weren't like specifically their own thing, but they were hyper specified in enough. Like usually this meant more like 50% of their genes outside of the core set were different from the other ones, but they were still enough of the core set to be considered some of the sample reference points. Does that make sense to everyone? I can't tell if people aren't talking or if I've been lost. Hello?
I don't really understand, but
It has to do with the spread of diversity. What's the best way to pull this up? So if you want to, oh, actually, you know what? Let's go to the next picture. So what you can see here, and this is where it gets interesting if you're into computational stuff, but you'll notice that that phylogenetic tree is like very fuzzy. It has a tremendous amount of lines, and you're seeing a lot of like piles of density in different ways, and so, and what you can see there are the five different groups that they pulled from, and then the individual groupings, the accessions within, right? All the different variations, and so and so you get the 3-letter code with the country of origin, and then your reference genomes. So these are the ones that are like the one that we're using as our model for this group, for this clade. And then here, the right hand graph here is all of the different laterally acquired genes, and then they're just letting you know here that they're connected by a vertical line, just so that like if there's gaps in between the two, you can see them there. The line is not a data point. And then they're ordered by abundance over groups, just in case somebody was to look at this and wonder why all the genes clustered on one side. It's because they've been reorganized by abundance. And for those of you who want a closer look, there is a picture of that. And does that partially go back and kind of explain what you were maybe a little curious about there, Mips?
I'm sort of confused about like how this thing works still, because I don't know. I've never seen this before.
Okay. Well, so right now what they're doing is clading, and so this is about, well, you've seen like a phylogenetic tree before, right? Like a tree of life or something akin to that, right?
Yeah, I think Taylor put it
Yeah.
up.
And they're
Yeah.
usually but they usually
Mhm.
look a little bit more linear, like less fuzzy. And you know, like they'll be like, here's the line, and then here's
Yeah.
the branch where like lizards comes off, and here's the branch where birds comes, right? Like all that kind of thing. This is just a very, very in-depth version of just one collection of
Yeah.
plants and
Okay.
the variations between them. Because when you're looking at those macro scales where you're like, well, you know, like here's where humans branched off from this line, and here's where
Okay.
dolphins happen and stuff like that.
Yeah.
Like those all have to do with genetic variation,
Okay.
right? And so within
Okay.
a
Mhm.
species, you can have clade and accession and stuff like that. And so you also have your own variety of genetic variation within a species. And so this this map that we're looking at right now is talking about that genetic variation within the species, specifically in regard to the lateral genes, genes that it's gotten from other grasses
Okay.
through non
Okay.
biblical methods. I don't know, like in the improper fashion. Is that a thinking?
Uh, are you going to go into how this is done?
You know, I
Okay.
totally am. But only a little
Wonderful.
bit, because it's mathematically
I
complex, but
think
I
you
do
cut a
have
little bit again.
yeah, you're clipping a little bit too, Mips.
Hold on.
All
Apologies.
right.
Oh,
Yeah,
okay.
let's keep going. Yeah. So, how is this done? So this is done by a tool called Beast, explicitly or specifically Beast 2.5. So Beast stands for Bayesian Evolutionary Analysis by sampling trees. And the like up until like Beast 2, it was a very standard thing, but then we kind of hit this point where the knowledge about how we're mapping these things out needs to change, right? Because like the concept of a very linear progression of genetics in a reality where most of the organisms are like hot swapping genes like mixtapes on a very regular basis, it gets a little blurry. And so this latest edition of Beast, it like really cranks up the blurry. And so actually up here in the right hand corner, you can see like, I know it's just like a screenshot. So, but the link to the paper is there, and you can and check it out. But those are all the new dependencies, and so a lot of this has to do about just taking in and handling more data, and then using non-tree and complex phylogenetic phylodynamics, right? Which is super cool. There's this whole thing in the paper that talks about extended phylogenetic structures, where they're like, hey, here's like a very standard phylogenetic structure, and that goes back to what we were just talking about, where it's like there's a line and a branch and a line and a branch, and then they're like, okay, yeah, but like, what about when you have things like coalitions of species or consortia or stuff like that, right? Like in entire microbiomes. Like I want to be able to map, like, because there are genetic independencies between things in microbiomes, especially when you have organisms that are using each other's metabolites to create a complex system. And so they kind of go into all of that stuff and talking about how to start envisioning that data. And that is the type of that is the software, and kind of like they're really excited about this. And if you look at other more Dunning papers, they like took this and kind of like tore this whole concept wide open. So it's been a very fun exploration. It's a little distracting, actually.
One question.
Mhm.
Oh yeah, so the Beast program, did that came out of the studying this lateral gene transfer thing, or did it like
Oh
is it
no, no,
just
no,
one of the
no!
new?
It's not even new. It's an existing piece of software that is used for this stuff. It's the combination of
Oh,
it's the combination of our understanding of that, and then that being applied to the software to expand the capabilities of the software, and then taking that and reapplying it, in kind of like a feedback loop. And like, and they're like, and then this is what we've gained, and it's like cool plant gene stuff.
oh, okay, I got you.
Mm-hmm.
That's the Beast
Yeah,
2.5, the where the 2.5.
yeah,
Okay,
exactly.
I see.
Where they're like, this allows us to really kind of explore some stuff that we've never really explored before. Not in the way that anyone's wanted to. Um,
Okay.
and yes, so there's actually not a tremendous amount of math at all in the paper, even in the supplemental, just because it's kind of more like we have this really powerful toy and we have done this thing with it, and and so they don't really go into that aspect of it. They're more interested in this, and this is kind of like one of the big takeaways from the paper, which has to do with these rates of gene transfer and the loss of laterally acquired genes, because in the same way uh, I was just having a conversation with somebody about the difference between uh, endogenous and exogenous chemicals, uh, like dopamine triggers and stuff like that, and the difference in how like your own internal chemical structures, and then externally derived from another thing, like a plant extract, for instance, has a much different effect, even though it's technically still working on the same pathway, like screwing with the same mechanisms. But so you have all of these lateral genes coming in, and they're not getting absorbed. And so what you can also do is you can, I mean, they are getting absorbed, but they have a very steady and calculable decay rate over time. And so that's the elapsed time. And so like phylogenetics is cool, but it's not my, like super focus. And I haven't actually looked at it. So it took me a while to remember what MA stood for. But that's the unit of time. It just means a million years. So yeah, so that's what's going on, this very calculable decay or loss of the gene. And then of course they do have like some instances are basically like what they call ancient laterally acquired genes, which are like things that are still there after like 10 million years or stuff like that. And then one of the things that this was kind of one of the big points that they were trying to get to, is that the genes get acquired at a specific rate and they decay at a calculable rate, and you can map the movements of these things and like see the history of the plant as it's moved genetically through time, what it's interacted with. Like historically, you can kind of link certain things to trade routes. There's a bunch of really cool stuff there. So this is like a very important piece of the basic cartography of the movement of genetics through time. Any questions so far?
This could probably be used to tell you the history of an organism over time.
So this specific, like the technique or the techniques used to get to where they got with the research could be applied to a lot of things. This is even, yeah, like you could see in a grass, for instance. So like if we do go to the next slide. This is the most gene-rich laterally acquired fragment. So they're like, this chunk right here has like all of these genes, and they have definitely come from these other ones, these ones that are not right next to them, that like that aren't even them. Like Zia maize, that's corn down there. There's sorghum. Like these are straight up grasses, and so you can see these conserved chunks, like the black bar up there is the gene for the ZAM sample, which they mentioned a lot. The ZAM sample had one of the most unique hybrid kind of weird ways of, like, it was the gene swapping king. It even had like within a field, you would have clusters of plants that were for whatever reason, it looked like they were kind of purposely clustering with other plants to increase option for transfer. Probably, like a very reasonable evolutionary trait in terms of spacing. But yeah, so yes, you can kind of see this stuff, and if one wanted to, you could start to follow the age of these genes, which they do have here. That's the well, not the age. They talk about it in another place where you can kind of map that. That's supplemental material stuff. All of which will be in the library, of course. but they talk about how you can map the age of the gene, so you can tell like how many million years of it's been around, and then you can see like colony like duplication amounts, like how often a gene is transferred. And so using that, you could, yes, kind of start to map out a little bit more of the complex system of how those things have moved through time. Not a gene specifically, but the clusters. Does that answer the question?
Yeah, I mean, and you can tell how they're different based on like that. Like an organism prefers a different like code. It prefers T's and A's over like C's and G's or something. Like that's probably how
Well,
they're seeing it.
this. I mean, like, are you asking how they tell they're lateral, or like if you
Yeah,
have a gene,
like is there like
if
a
you have a
rule?
gene, if you have a gene here and you have a gene here, like how do you tell that those are laterally as opposed to like nuclear or like conserved genes, right? Is that the question?
Yeah,
Right. And so
basically, yeah.
some of that actually like can be done with time. Like you can see where that gene came through. Like if the core model of the organisms has like this cluster, and then after that other genes which haven't evolved in that thing, which definitely evolved in something else, right? Like you can follow the line. If the line for so like let's this graphic will be of no use, but like if you have the line as the progression of the gene through time, and it is moving along the track that says corn, right? And at some point, that gene, that line is suddenly like forked and is also in sorghum. There's not a lot of oh, it just like magically created this complex gene set which just happens to look like this other complex gene set which is already pre-existing, when they happen to
Oh,
just be smashing into each other like in physical real time all the time, right?
ah,
Yeah, and so
hmm
you can
hmm
map that, and there's a whole section again in the supplemental where they talk about like the specifics of what defines a lateral gene, especially within the confines of this study, you know. And so it's like, okay, so it's got to be like two steps removed, but then, or no, it's got to be like there's a shift point and then it's surrounded by enough sister clones that it can be shown that the gene came from this, which was a transfer, as opposed to like kind of a hybrid-y, mutation-y type of stuff. Because if it was vertical transfer, it would be somehow a cut and paste job from the pre-existing genome, right? Like you can't get entirely new
hmm
paragraphs from, like, if you've got two copies of Time magazine, like you're never going to find a Rolling Stone article in the baby of those two copies of Time magazine, right?
right. Okay,
Yeah.
so this is this. So the so like a large like a variant population is needed for like figuring this out too.
Yeah, absolutely.
Okay,
Because I
okay.
mean,
So it
because
wouldn't be as
otherwise,
useful for like living fossils,
um,
like monotypic genus things, like right?
oh, so I'm going to say that this particular instance of how the technology is used does not mirror over, but that doesn't mean that you can't take pieces of this and the tools and do other cool stuff a lot more in line with that.
Okay, yeah,
Yeah.
very interesting.
All right, where are we at? Oh yes, we were talking about the most laterally rich acquired fragment. And I think we talked about that quite enough. And then there were a couple of really big takeaways, and I'm going to have to apologize. Like, I actually just flaked out and thought I had completed this slide, and I didn't. And, I'm just going to have to own that because that's not even a properly formatted block of text. But that's okay because I can talk about it. So the takeaway of this paper, kind of like, and you can go back and look at the title of the paper, which is about this kind of extra, like the movement of genes through time, right? Genes that accumulate at different rates within a grass lineage. And so one of their kind of big takeaway points is that, like, this is not just chaos. This is an evolutionary adaptation. This is how these grass species maintain their hybrid vigor, how they deal with new situations. They talked about how you would have things like blights or plagues that would come through and push genetic change in the survivors of the grass, and then those genes would actually be transferred laterally to other species. And then as that blight or as that season of whatever, not season, when I say season, again, we're talking millions of years here, but like, so as the time moves on and the stress is gone, the gene, which is these lateral genes are kind of, they're predominantly what are considered like accessory genes. They're not like the core fundamental stuff. You don't get a lot of the lateral genes coming in and then getting locked down indefinitely. Like you saw from those graphs, like, you hit like 5 million years or something like that, and you're about 50% down with anything that's been acquired because it just isn't useful anymore. It's been a long, long time. But there is an equal and calculable rate of things coming in all the time. And in the same way that bacteria are transferring plasmids to maintain their hybrid vigor, we're seeing this very steady cycle of genetics moving through the species laterally so that they can maintain that type of thing as well. And that is said way better than whatever garbage I put there. Yeah, so that's like the basic outline of the paper as it is. As I said, all of the papers that I referenced and the supplemental material be tossed in the library. Questions?
Okay. So yeah, I had one. So like, with this paper and like the conclusion you just said, like, how would this change our understanding of like evolution of like this grass species? Like, does it fill in any holes or like things we didn't understand before?
It adds, I think what I would say is that it adds like a layer of complexity. And not in a bad way. It just broadens our understanding of how this works.
Okay.
There are some parts where they talk about like how this lateral gene transfer happens and how it resembles certain techniques that we have. And then, like, they look a little, so we're learning more about the tools that we develop by watching them be used in the wild, because all of our genetic tools are biology's tools to begin with.
Yeah.
So in terms of that, but also it just like it really is a good paper, and especially with all the extra stuff around it, it really feels like a solid piece of information where they're like, so this
Yeah.
is kind of a thing now. We're going to need to readdress certain things. It's not going to like break anything. This isn't going out there and shaking the foundations of
Yeah.
sixth grade biology, but it's very cool, and it's I think that the what they've done, the how they've done it, like this has a lot of potential, because like if this can be kind of, if they can take this and codify this and slide this sideways from grass, like this is going to really expand how we look at things, and I think that's really cool.
Yeah. Mm-hmm.
Yeah. Any other questions?
I'm looking it up a little bit right now, and I see something about pollen to pathway transformation. Like that seems like
Yeah.
like you could just insert like a gene directly into the pollen tube, like
Yeah.
hardly any
Which
like
is
like
that's a thing. That's a
wow.
thing. That's a thing. Just like stick it in there, right?
Yeah,
Yeah.
it seems like really useful. That'd be a really cool way to experiment in modifying. Like I wonder
Mhm.
how like it says pollen tube transfer. I wonder if like
I
the
mean,
less
in
protected the ovule is, the more gene transfer occurs. I would guess. Like
in all fairness, we have
you know what I mean?
some pollen dipping techniques for plant transformation that are
Very
pretty
interesting.
lazy. Where it's just like, so you take the flower and you're just gonna
I can
stick
barely
that
hear you.
flower. Okay, sorry about that. I don't know how to fix that, but basically, we've got these techniques where it's like it's called pollen dipping, where it's like you just stick it in the bacteria, just go, just blorp, or collecting. Like we do a lot of collecting pollen, and then you can actually like load stuff on the pollen, which is cool. All right. Um, if there aren't any more questions, I'm going to stop sharing the screen. And thank you all for coming to Journal Club.
Thank you, Gabe. That was a really cool
Yeah.
paper, honestly.
Yeah, I mean, I like that. That was some like weird plant stuff,
Yeah.
Could you repeat what you said? I think I lost connection.
Which part?
Right after the pollen tube thing.
Oh, yeah. Evidently, I got lost a little bit there. Yeah, I was talking about pollen dipping. How we basically have plant transformation techniques that are just like, have you considered just smashing it, like just smush it on there? I don't know, just rub it in bacteria. Like, I
Whoa.
mean, well, think about it. At every level, it is still physical reality, and that just means you need enough small objects bonking into each other.
Yeah.
I mean,
You probably don't
that's
even
how like all genetic work happens.
right. You probably don't even need like a gene gun or anything. You can just put it in a solution or in the pollen and
Pollen,
put the
pollen, pollen dipping is a thing for sure.
yeah. That
Yeah,
sounds absolutely cool.
gene guns are kind of like a go-to thing. One, because they're very easy and consistent once calibrated. If you're doing industrial work, pew pew pew, it's magic.
Right.
But the other thing is that until just recently, like our control and restriction on how we use agrobacterium has been a lot tighter in this country. And so if you're developing technology for it, and we are developing that technology, then you needed to have something that didn't necessarily make it so that when you were doing your research, or if you wanted your research to be duplicated, or if you wanted your method to be duplicated, you found the method that was the least litigiously contentious. right? And that was Gene Gun. Like it's very hard to say, yes, I smacked it with an air rifle, and they'll be like, oh, well, don't do that. It's like there are limits. But now our agrobacterium control is lower than it used to be, which, in all fairness, I don't see as a lapse in judgment of the regulatory organization, just because, like, it's a fucking soil bacteria. You're not gonna like, you can't ship it across state lines. It was such a stupid rule. Anyway, all right, so I'm gonna stop the recording here with Craig.